Go ahead and title your notes Photosynthesis. And similar to last time, we're drawing a large cell, and we're going to show everything that occurs inside that cell. But let's start with the equation, which shows us our inputs and outputs. One of the molecules that is required for photosynthesis is carbon dioxide. We will also need water. And then the end product, of course, is a sugar molecule, specifically glucose. And then an oxygen will also be a byproduct of this reaction. Go ahead and draw a large rectangular cell. Rectangular because it's going to represent a plant cell. And so we're going to have something like this. I'm sorry. Cancel that. Gosh. I forgot. We're doing this is not an image of the cell. This is an image of the chloroplast. And I need to make sure I always forget that. So this is actually roundish because this is not a cell. This is actually an organelle that we're looking at. So label it chloroplast, okay? And the reason that this organelle is shaped like this is because you recall that the chloroplast, it used to be a bacteria. It used to be a eukaryote, and it became engulfed, and it started living in a symbiotic relationship, became an organelle. So the chloroplast has its own DNA. That's just one of the evidences that it used to be a prokaryote. Okay, now within the chloroplast is, I don't know if you remember it looked like a stack of coins. It was called a thylakoid. We're going to take a look at one of those thylakoids. And you're going to have, it's going to look kind of like this. So think of it as like a hollow disc. And so this would be the thylakoid membrane. It's got some thickness to it. And then inside the thylakoid, the structure would be the thylakoid space. So you've got this cavity. It's like a hollowed out structure, and inside is this cavity. And there's going to be something very familiar to us that we saw last time. There's going to be this buildup of protons again. So you're going to end up, the reaction that we do is going to end up producing a massive buildup or a concentration gradient of protons. So proton accumulation. Proton, proton, proton. And so the question is, now you're good. What do you need?
Oh, I was going to show you something, but I'll do your first.
All right. Okay, so the question is, how do we get this accumulation of protons? So let's start with photosystem 2. So there's a protein embedded in the thylakoid membrane called photosystem 2. And the photosystem 2 protein is pigmented. So we're drawing these dark pigment granules. And anybody know what these dark green pigments are called? Chlorophyll, yeah. So we've got light that's going to strike the chlorophyll pigments. And those chlorophyll pigments are going to absorb that light's energy. So you can say that this is light coming in. The light strikes the chlorophyll, and then something's going to happen. And the thing that's going to happen is an electron is going to get excited. And all we mean by excited is that it's going to jump up in energy. The electron receives the energy and gets excited to a high energy state. Okay, so the question though is, where are we getting these electrons? Because we're just going to keep exciting electrons. We must have a source of electrons, and that source is water molecules. So the water molecule is going to provide the electron. It's also going to split into an oxygen molecule, and protons. So now we understand where we're getting. We understand a couple things actually from this reaction. We understand where the protons are coming from, right? They're coming from the water. We now understand where oxygen gas is made, right? That's also coming from the water splitting. We call it water splitting. And then we also know where these electrons are coming from, right? It's also coming from the water molecule. So this reaction is called the water splitting reaction. It also has a fancier name called photolysis or photolysis. And if you break down the etymology of the word, photo means light, and then lys means split. So photolysis, also known as water splitting. And the overall reaction, I'm just going to sum it up over here. We're going to say that 2 water molecules will yield 1 oxygen atom, 4 protons, and 4 electrons. And so this right here is also shown right here. It's the water splitting. So we can actually check off a couple things from our master equation at the top. We can check off oxygen as an output, right? We've now shown how oxygen is produced. We've also shown how water is consumed, right? So we've shown we've shown an input and an output already, just in this short little explanation. Unfortunately, there is more to the picture, so we're going to keep adding here. So the next step is that there's also photosystem 1, and it looks similar. It's got chlorophyll. And so again, you got the light coming in. And basically what will happen is the electron will get further excited. So the electron gets excited again by photosystem 1. So this is Roman numeral 1. This is Roman numeral 2 photosystem. So why does photosystem 2 come first? Why does it? I don't know. I'm not sure. But anyway, you get the electrons get excited. And so remember last time how we like to trace the energy? We like try to follow the energy and where it's going. What we've got energy coming in. So the energy's coming in, and now it's transferred to the electrons, right? So the energy's been transferred from light to electrons. So what are these energized electrons going to do? They're going to work with a enzyme called NADP plus. Sorry, I wrote that kind of sloppy. Let me fix it. NAD plus reductase. And what do we know this thing is? It ends in the letters ACE. So it is a enzyme. Yep, it ends in ACE. It's an enzyme. And specifically, what it can do is it can energize the energy carrier. So we've got an energy carrier NADP plus that's going to join up with this electron. So they're going to come together. So you got the electron, and then you got the NADP plus a proton. And the proton is in solution already. But if you get these three things coming together, you got an electron, an energized electron, a proton, and an NADP plus, you will get an NADPH. And this is our energy carrier, right? We've seen it before. It's utilized by cell respiration as an energy carrier. And so it's gonna do just what its name suggests. It's gonna carry the energy. And it's gonna carry the energy over to the stroma where we're gonna build the sugars. Okay, now one of the other things that's occurring is we have, once again, we've seen this molecule before as well. It's called ATP synthase. So you guys, a lot of this is repetitive. A lot of the same stuff is happening here that was happening in cell respiration. So the ATP synthase is also it's going to let out the protons, right? The protons are going to flow through, and ADP plus a phosphate will become ATP. So there's going to be a production of ATP as well. Now, ATP is also an energy carrier. It's a type of energy carrier. So we've got two energized molecules now. So again, let's trace the energy. So now the energy has started here. It got transferred to the electrons, and then from the electrons, it got added to these energy carriers. So right now the energy is right here, and we're going to go and do something with that energy. Any ideas what we might do with the energy? Think about photosynthesis. What are we trying to make here? Nobody knows? Or do you just not, you guys just don't want your voice to be on the videotape because we're recording? Yeah, all right. That's fine. It's just, yeah. So we're trying to make sugar, right? We're trying to make sugars. But a sugar molecule it takes quite a bit of energy to make one because you have to connect. It's an anabolic reaction, which means you're building up, and that means it's going to require energy to build it up. So we're going to need this energy to build the molecule. All right, so label this as Calvin cycle. It is occurring in the stroma of the chloroplast. Okay. So the stroma is just kind of like the solution that's in the main space of the chloroplast. So we got the Calvin cycle. So let's go ahead and start with this thing called this thing called something that I forget the name of. I'm sorry, you guys, I'm blanking on the name, but it's a 5-carbon molecule. So picture 5 carbons connected together, and then it's capped with some phosphates on the end. And this little molecule is going to react with Rubisco, abbreviated RUBP. It's just an enzyme that's going to help process it. And it's going to react with a carbon dioxide molecule. 2, 3, 4, 5. So picture a carbon dioxide molecule coming in, reacting with Rubisco and the compound here, and then producing 1, 2, 3, 2 different 3-carbon molecules. So if you do the math here, if you count the carbons, so we've already established, right, that our little circles, they represent carbon atoms. And if we have 1, 2, 3, 4, 5, plus the 1 carbon atom from here, you got 1, 2, 3, 4, 5, 6, and then we've got 1, 2, 3, 4, 5, 6. So we've accounted for all the carbon atoms. There's conservation of matter here. I just want to prove that. I want to show you that conservation of matter. Okay, so one of these 3-carbon molecules will react will react to form glucose. So this is going to produce a glucose molecule. And you're going to need 2 of them. Times 2. So put a little times 2 right here. And then the other thing we need to build this sugar molecule is we need energy. And so thankfully we've got the energy. So show with a dotted line, some of this ATP is going to come down here. And you're going to have ATP going to ADP. So anytime we go from ATP to ADP, that means that we are spending energy, right? Or another way of thinking about this is that this energy is being added to the compound to make sugar. So the energy is going from here to here. Okay, so next, the other molecule will go through a process called RUBP regeneration. And all that really means is that there's a cycle, there's a cyclic thing going on here. However, the regeneration also requires ATP. And so you get this ATP going to ADP again. And then the cycle repeats. So does everybody see how this thing just goes on and on and on? Which means that it also means that we need a constant input of ATP. We need a constant and perpetual source of ATP. We also need a constant source of carbon coming in. And then from there, we will have a constant production of sugar. As long as we have a constant source of light, because that light, if you really go back to the very beginning of this process, the light is really the first and foremost source of all of this energy and everything that's happening here. Now, what about the energy carriers? I forgot to show you what happened, the NADPH is also utilized in this reaction. So we can also put NADPH becomes NADP plus. So squeeze that in there. That's also occurring in this step. When the carbon compound is turned into a sugar, we consume ATP. We also consume NADPH. I forgot NADPH. NADPH. Okay, I know it's complicated, and or NADP plus. I know it's complicated. I know that you're looking at this and you're thinking, okay, gosh, like, what am I supposed to like memorize this diagram? What do I? No, the answer is no, because there's never gonna be. You're just never gonna be asked to replicate the diagram, right? But you will be asked questions like, where where is the energy transferred to energy carriers, and you'd need to be able to say that's occurring in that's occurring with the thylakoid. If you got asked questions about where is carbon fixation occurring, you'd need to be able to say that that's occurring in the stroma, right, with the Calvin cycle. So you're going to get asked questions, and so if you have this diagram kind of in your brain a little bit at least, you're going to have a much easier time picturing in your mind like where stuff is occurring, right? And you're going to get some practice problems too before Friday. I'm going to give you guys some sample problems that we're going to look at, and then you should be right now on your own time. You should be looking at some sample problems from the AD classroom and kind of getting a feel for what kind of questions you might be asked about photosynthesis. So, any questions about the diagram? Okay, one last thing to finish the thought here. We have water coming in, right? So we need to check off the remaining inputs and outputs. So if we go up to the master equation, have we accounted for carbon dioxide as an input? Yes, over here. Carbon dioxide input, check, check. And lastly, have we accounted for sugar as a output? Check, check, right here. That's where it's made. So we're good. We already went over the water splitting. We showed that process over here. Okay, the only other thing to consider that's kind of interesting is that there's sort of this compatibility. There's sort of this like the word I'm looking for is complementarity. So imagine you've got a mitochondria over here. The outputs of the mitochondria are the inputs to the chloroplast. Did you guys ever notice that? Kind of the complementarity. So we know that water is produced and released by the mitochondria. We know that carbon dioxide is also released. And then one of the things we know is consumed by, and by the way, we already have a diagram of this. We have a diagram of cell respiration. We also know that the very sugars that are made by the chloroplast are are the input for the mitochondria. So that's why we call plants, that's why we call them autotrophs, which means self-feeding. So the plant in its chloroplast, it'll make a sugar molecule and then it will feed that sugar molecule to itself. It will literally feed it to itself. It'll give that sugar straight over to the mitochondria so that it can make ATP. Kind of cool, right? This complementarity. Furthermore, this what is the other input for cell respiration? Aerobic cell respiration, I should say. Oxygen, right? And lo and behold, the oxygen molecule up here can also feed the process of cell respiration. Got your oxygen molecule coming in. And then this CO2 will feed the Calvin cycle because we know that that's an input there. And then we also know that water is an input here. And so you see the complementarity, you see the cycling. So students, this is why you need to have just as many mitochondria as chloroplasts in your mesocosm. You need this cycling to be happening, right? You need it to be in balance so that the carbon is passed around and so that the energy is passed around. There's a cycling going on there, right? Okay. Questions, comments? Good stuff. So I'm going to stop the video